Welcome to the deep dive. Today, we're diving into something that honestly feels like it's straight out of science fiction, but it's very real. Gene editing.
Yeah, specifically CRISPR-Cas9.
Exactly, CRISPR-Cas9, a tool that's just fundamentally reshaped our ability to, well, to modify life itself. You've sent us a really fascinating pile of sources here, everything from deep scientific reviews to the very latest clinical trial updates.
Mm-hmm.
And our mission today is basically to distill the most important stuff, the key knowledge and insights from all this for you.
Such a dynamic field. I mean, the advancements just in the last 10 years or so,
Huh,
they're breathtaking, really.
absolutely.
So we should probably start right at the core.
Hmm.
What CRISPR-Cas9 actually is, where it even came from, which is kind of surprising,
Right.
and, how it works at a really fundamental level. Because once you get that, you really start to see why it's so powerful.
Totally agree. We're talking about tech that's already changing, well, everything. How we treat diseases, how we grow our food.
Yeah, even how we think about human life itself.
So, for someone just jumping into this deep dive with us, what is this groundbreaking tool, like in simple terms?
Okay, so at its heart, CRISPR-Cas9 is basically this really sophisticated defense system. It comes from bacteria.
Bacteria, really?
Yeah, imagine like a tiny immune system inside a bacterium that remembers viruses that have attacked it before.
Huh. Okay.
So when it sees that virus again, it uses these tiny, precise molecular scissors to just chalk up the virus's DNA, kills the threat.
Wow.
And that's essentially the mechanism we figured out how to borrow and use.
Okay, let's unpack that. Molecular scissors. How do these scissors know what to cut? I mean, a genome is huge and complicated.
Right, good question. It basically uses two key parts working together.
Okay,
First, there's the Cas9 protein, that's the actual scissor part.
got it.
And second, there's something called a single guide RNA, or sgRNA.
sgRNA.
Think of that sgRNA as like the ultimate GPS. It's programmed with a sequence that exactly matches the bit of DNA we want to target.
Ah, so it leads the scissors to the right place.
Precisely. It guides Cas9 right to that spot. Once Cas9 gets there, it makes a very clean cut across both strands of the DNA helix, a double-strand break.
Okay, so it cuts the DNA. Then what?
Then the cell's own natural repair crew kicks in. Cells hate having broken DNA.
Right, they try to fix it.
Exactly.
Oh.
And we can kind of strategically use those natural repair mechanisms to introduce the changes we want, maybe fix a faulty gene, delete a bad sequence, or even insert something new.
That's ingenious.
It really is. And this whole system, it was first noticed way back in 1987 in E. coli. But it wasn't until 2014 in this huge paper in Science by Jennifer Doudna and Emmanuel Charpentier.
I remember that making waves.
Huge waves. They showed how to actually harness this system outside a cell, in vitro for targeted DNA cutting. That just opened the floodgates.
I can see why. The sources you sent really hammer home how its simplicity, its low cost, and just how well it works.
Yeah.
It was a total game changer, a paradigm shift.
Absolutely.
But stepping back, before CRISPR, trying to edit genes must have been completely different, right?
Right.
Much harder.
Oh, incredibly different. I mean, people wanted to edit genes, especially in stem cells, to make model organisms and stuff for decades. But the methods, they were just extraordinarily difficult.
Like what? What did they even try?
Well, really early on, you had things like just blasting cells with radiation or chemicals to cause random mutations.
Random. That sounds messy.
Totally messy. Or using viruses, often imprecise, like throwing darts in the dark. And often the changes wouldn't even be passed on one-off experiments.
So what was the first real step towards something more targeted?
Well, a big turning point came in the early 80s with mouse embryonic stem cells, MESCs. Scientists like Martin Evans and Gail Martin figured out how to work with these.
Okay, stem cells from mice.
Right. And that meant you could make genetic changes in a dish in vitro and then use those cells to create genetically modified mice. That work, plus contributions from Mario Capecchi and Oliver Smithies on something called gene targeting.
Gene targeting. Based on homologous recombination.
Exactly. Using the cell's natural DNA swapping process. That whole body of work actually got them the Nobel Prize in 2007.
Wow. But it still wasn't easy.
No, not at all. That homologous recombination process, notoriously inefficient, super laborious, incredibly expensive.
Yeah.
You'd literally start with millions of cells
Millions.
and maybe get a handful that were correctly modified. And even then, you constantly worried about accidentally changing something else, off-target effects.
Good grief. So a huge amount of effort for potentially tiny results. You can see why they needed something better.
Desperately needed something more precise, more efficient.
And that led to the idea of targeted nucleuses, these
Yeah.
engineered scissors.
Precisely.
Mhm.
So the field then moved towards things like zinc finger nucleuses,
Yeah.
ZFNs, in the late 90s.
These offends.
And then later, in the late 2000s, Talon's transcription activator-like effector nucleuses.
Okay, ZFNs and talons, how did they work?
ZFNs were sort of the first generation of site-specific editors. They worked by fusing these specific DNA-binding proteins, the zinc fingers, to an enzyme that cuts DNA. They even got used in some early clinical trials, actually,
Oh, really? Like for what?
trying to disable a gene called CCR5 for HIV treatment, for example.
Interesting. And talons.
Talons came next.
Yeah.
They were generally more flexible to design and maybe a bit less toxic than ZFN, so they got pretty popular for a while.
But still not perfect.
Still challenging. Designing and building these custom proteins for every single gene target was complex, time-consuming work.
Okay, so that sets the stage. What was the big leap? What made CRISPR-Cas9 just blow past even ZFNs and talons and become this huge phenomenon?
Right, here's the key thing. Unlike ZFNs and talons, where you had to engineer a whole new complex protein for each target,
Right, custom proteins.
Cas9, the scissor protein, is always the same. It's guided to its target by that simple, easy-to-make, programmable RNA molecule, the sgRNA.
So you just need to change the RNA guide and not rebuild the whole protein scissor.
Exactly. That made gene editing dramatically more accessible, faster, way cheaper.
So labs everywhere could suddenly do it.
Suddenly, thousands of labs worldwide, not just a few specialized ones, could jump into high-precision gene targeting research. That ease of use is why we saw this just boom, exponential explosion in papers and discoveries after 2014. It democratized the whole field.
Okay, so CRISPR-Cas9 itself was revolutionary. But science never stands still. What's happening now? Are people just tweaking CRISPR, or are there totally new things coming along?
It's really both. On one hand, yes, there's a lot of work refining CRISPR-Cas9 itself.
How so?
Well, like developing high-fidelity versions of the Cas9 scissor, things like ESP-Cas9 or HypaCas9, that are much less likely to make those accidental off-target cuts.
Making it safer.
Right. And also using things like machine learning, AI, to help predict which guide RNAs will work best and be safest for a specific target.
Okay, so improving the original. But you said new approaches too, beyond CRISPR-Cas9.
Yes, absolutely. The field is moving incredibly fast towards tools that are even more precise, sometimes more versatile.
Can you give us some examples? What are these next-gen tools?
Sure. One really significant development is called prime editing. This came out in 2019 from David Liu's lab, work led by Andrew Anzalone.
Prime editing. What's the concept?
It's often described as like a search and replace function for DNA, almost like a word processor.
Okay.
It cleverly merges the targeting ability of CRISPR-Cas9 using a modified Cas9 that only nicks one strand, not a full cut, with an enzyme called reverse transcriptase.
Reverse transcriptase, like from viruses.
Exactly. And this combo allows it to directly write in new genetic information at the target site without needing that double-strand break and without needing a separate DNA template to copy from.
So no big cut, just rewriting.
Pretty much. It can make all 12 possible single-letter swaps, plus small insertions and deletions. And because it avoids the double-strand break, the risk of unwanted mutations seems to be lower.
That sounds incredibly precise. How powerful is it?
Well, the sources we looked at suggest it has the potential to correct something like 89% of known disease-causing genetic variants in humans.
89%. Wow, that's huge. What about base editing? I've heard about that too.
Right. Base editing came a bit earlier, around 2016, also largely from David Liu's lab, work by Alexis Calmore and others. It also avoids making double-strand breaks.
Okay, another approach without the full cut.
Yeah. It uses a disabled Cas9 one that can find the target but can't cut fused to a different kind of enzyme, a base deaminase.
A deaminase.
This enzyme directly converts one DNA letter or base into another, right there on the DNA strand. Specifically, you can change a C to a T or an A to a G.
So very specific single-letter changes.
Exactly. Highly precise for fixing those single-letter typos in the genetic code, what we call point mutations.
So, okay, we've got the original CRISPR, which is now easier and safer, then base editing for specific letter swaps, and prime editing for search and replace. Seems like the toolkit is getting really sophisticated.
It really is.
But what does that mean for, the average researcher or eventually for patients? How accessible are these newer, fancier tools?
That's a really important point. While the basic CRISPR-Cas9 system has become pretty cheap and it's used everywhere,
Right, it's spread like wildfire.
prime editing and base editing currently need a bit more specialized gear, more technical know-how. So they haven't quite seen that same super widespread adoption yet.
So still more the domain of specialists for now.
For now, perhaps,
Yeah.
but there's a huge push happening to make these systems simpler, more robust, cheaper. The goal is definitely to get these powerful tools into more hands, labs, and eventually clinics worldwide. Democratization continues.
Okay, let's shift gears then to where this hits home for a lot of people. Human health, medicine. The potential seems just immense, especially for genetic diseases. What are we actually seeing happen now?
Yeah, this is where things get really exciting and, frankly, quite moving. CRISPR-Cas9 is being aimed right at diseases caused by specific gene mutations.
Like what? What are the big targets?
Well, some of the most promising work is in hereditary blood disorders, things like sickle cell disease, SCD, and beta thalassemia.
Okay, I know sickle cell, debilitating.
Absolutely. And both are caused by just single-letter mistakes, single nucleotide mutations in the genes for hemoglobin. That's the protein carrying oxygen in your blood.
Right, so a single typo causes all that suffering.
Exactly. And back in 2016, there was this groundbreaking preclinical study. They took blood-forming stem cells, hematopoietic stem cells, from sickle cell patients, used CRISPR-Cas9 to actually correct the mutation in those cells in the lab, and showed they could restore normal function.
In lab models, okay. But what about people?
Well, fast forward to 2021, and a clinical trial report by Frangul and colleagues. They used CRISPR to edit a specific gene, BCL11A, in patients' stem cells, which boosts production of fetal hemoglobin.
Okay.
And the results were remarkable. Significant improvements in symptoms. Transfusion needs basically eliminated for some patients. Real impact.
That's genuinely incredible. Life-changing stuff. What other diseases are in the crosshairs?
Well, researchers are definitely exploring it for cystic fibrosis, CF.
CF, that affects the lungs' digestive system.
Right, due to mutations in a gene called CFTR. It messes with how salt and water move across cells, leading to thick mucus. Now, delivering CRISPR to the lungs is tricky because of that mucus.
I can imagine.
But recent studies are showing progress. They've managed to edit the CFTR gene in human stem cells in culture, using things like lipid nanoparticles or harmless viruses like AAVs to carry the CRISPR machinery. And they've seen restored function in those lab models.
So progress, but maybe further from the clinic than sickle cell?
Perhaps, but moving forward, we're also seeing work in muscular dystrophy, Huntington's disease, even inherited forms of blindness.
Blindness, how does that work?
Yeah, there's a preclinical study in 2019 for a condition called Leber congenital amaurosis type 10, LCA10. They developed a CRISPR therapy called EDIT-101
EDIT 101.
and showed it could work in vivo, meaning directly inside living mouse models that were humanized, designed to mimic the human disease. And that actually moved into human trials.
Directly editing genes inside someone's eye? That sounds futuristic. How far along are these trials generally?
Well, the landscape changes fast. Our sources from 2016 mention about 12 trials using targeted nucleases, mostly the older ZFNs for things like HIV.
Okay, that was them.
Right. More recent data confirms these ongoing, promising CRISPR trials for sickle cell and beta thalassemia, and indeed the in vivo trial for that form of blindness. Early results are encouraging.
But there are still hurdles.
Oh, absolutely. Huge hurdles remain. Ensuring long-term safety is paramount, making absolutely sure we minimize those off-target cuts, and just figuring out the best ways to deliver the CRISPR system to the right cells in the body efficiently. That delivery challenge is a big one for many diseases.
And the elephant in the room, cost. These sound like incredibly complex, personalized treatments. Are they going to be affordable, accessible?
That's the multi-billion-dollar question, isn't it? And a critical ethical one. Right now, yes, the first CRISPR-based therapies are coming with very high price tags.
Which limits who can get them?
Exactly. It raises serious equity concerns. However, the hope and expectation reflected in our sources is that as the technology matures, as manufacturing scales up, as delivery gets better and cheaper, the costs should come down significantly over time.
We have to hope so. Making these cures available to everyone who needs them seems crucial.
Absolutely. It's a major focus.
Okay, so medicine is obviously a huge area, but CRISPR isn't just about human health, right? The sources talked a lot about agriculture and biotech, too.
Definitely. If you look at the bigger picture, CRISPR is a powerful tool for improving crops.
How so? What can it do for farming?
Well, think about modifying genes related to disease resistance, making plants less vulnerable to pests or fungi, or boosting yield, improving nutritional content.
So better crops, faster than traditional breeding.
Much faster and much more precisely. Traditional breeding can take years, decades even, and it's kind of a scattergun approach sometimes. CRISPR lets you make very specific changes.
Can you give some concrete examples?
Sure, things like creating rice varieties that are resistant to bacterial blight or fungal infections, or engineering maize corn to be more drought resistant.
Drought resistance. That's huge with climate change.
Exactly. There was a study in 2017 where they used CRISPR to tweak a gene called RGOS8 in maize. And under drought conditions, it increased grain yield by 5 bushels per acre.
Wow, that's a real difference.
It is. Or think about biofortification boosting vitamins or minerals in staple crops like wheat or potatoes, or reducing allergens or anti-nutritional factors in things like soybeans, making food safer and healthier.
What about the non-browning mushrooms or the tomatoes mentioned in the sources?
Right. Those are great examples of improving traits consumers care about, like shelf life or even health benefits. The tomatoes had increased GABA, which might help with blood pressure. It's also being used to make cassava virus resistant or to change the oil profile in soybeans to be healthier.
And livestock, too.
Yep, being explored there as well. Improving meat quality, disease resistance in cattle, pigs, chickens.
It really sounds like it could help tackle global food security.
The potential is definitely there.
And beyond food, what about other biotech applications? Biofuels, industrial stuff.
Big impact there too. CRISPR is used to engineer microorganisms like yeast and algae
For what purpose?
to make them produce more lipids, oils, which are key for making biodiesel, or engineering microbes to be better at breaking down plant material into bioethanol.
So greener fuels and chemicals.
Exactly. Making industrial processes more efficient, more sustainable, less reliant on fossil fuels, even engineering microbes that can survive really harsh industrial conditions, like high heat.
And fisheries.
Yep. Improving farmed fish through genetics, potentially controlling invasive aquatic species, enhancing fish health in aquaculture.
Okay, one more connection the sources mentioned. Nanotechnology. How does that fit in? Seems like a different world.
It might seem like it, but there's a strong synergy. Nanotechnology gives us incredible control at the tiny nanoscale level. So imagine using nanoparticles as super precise delivery vehicles. Tiny packages designed to carry the CRISPR components, the Cas9, and guide RNA directly to specific target cells in the body and nowhere else.
Ah, so solving that delivery problem we talked about earlier.
Potentially, yes. Or using nanoparticles for imaging, to actually watch the gene editing process happen in real time inside a living organism. It's about combining precision targeting with precision tools.
Okay, this technology is clearly incredibly powerful with applications everywhere, which inevitably brings us to the really big questions. The ethical, legal, social stuff.
Yes, this is absolutely crucial. The power of CRISPR forces us to confront some very deep questions.
So what are the main ethical hotspots?
Well, while benefits are clear, CRISPR has definitely sparked huge controversy. A major debate is around germline editing.
Germline, meaning changes that can be passed down to future generations.
Exactly. Editing the DNA of sperm, eggs, or embryos. This raises profound issues about, unforeseen long-term consequences, altering the human gene pool.
And the whole designer babies fear.
Right. The concern that we could start selecting for desirable traits, leading to a kind of genetic enhancement race, creating societal divides.
I remember the sources mentioning the Chinese twins case back in 2018. That really blew up, didn't it?
It absolutely did. The announcement that twin girls had been born whose genomes were edited using CRISPR before birth, it caused international outcry.
Understandably.
It massively intensified calls for strict rules, global oversight. The ethical worries are huge. Potential for new forms of discrimination, unfair advantages for the genetically enhanced. It's a minefield.
And the ethics are just about humans, right? What about editing the environment?
Another huge area of concern.
Yeah.
Especially with technologies like gene drives.
Gene drives designed to spread a genetic change through a whole population.
Precisely. The idea is to say, make all mosquitoes incapable of carrying malaria and then have that trait spread rapidly through the wild population.
Which sounds great in theory for fighting malaria.
The potential public health benefit is enormous. But the ecological risks. What happens if you accidentally wipe out a species or drastically alter it? The downstream effects in the ecosystem are incredibly hard to predict, could cause major imbalances.
So immense power again, demanding extreme caution.
Extreme caution, thorough risk assessment, robust regulation. Absolutely essential before we even think about deploying something like that widely.
Speaking of regulations, how is the world actually governing this? Is there any global consensus?
It's patchy, very diverse.
Yeah.
You've got international bodies like the World Health Organization, the WHO, putting out guidelines, emphasizing responsibility, transparency, talking to the public.
Okay, guidelines, but laws.
Well, in the U.S., agencies like the NIH and FDA have rigorous review processes, especially for clinical applications. But then you look at Europe,
Stricter, I gather.
much stricter. They generally classify gene-edited organisms, even plants, under their existing, very stringent GMO regulations, which some argue really stifles innovation there.
And China, after the twins incident.
They significantly tighten their regulations. So you have this real patchwork quilt of laws globally, reflecting different cultural values, different risk tolerances.
Which must make things complicated for researchers and companies working internationally.
Very complicated.
And beyond the laws, what about public opinion? And this issue of access and inequality.
Public perception is also really varied. You see cautious optimism, especially about using CRISPR for serious diseases, but also significant anxiety about safety, ethics, control.
And the inequality gap.
It's a massive concern. Right now, the cutting-edge research, the expensive therapies, they're concentrated in wealthier developed countries. There's a real risk that gene editing could widen the gap between the haves and have-nots globally.
Both between countries and within them, right? If treatments are super expensive.
Absolutely. That cost barrier we talked about could mean only the affluent benefit, at least initially. Addressing access and equity is a fundamental challenge.
Okay, so navigating this complex landscape. Where does CRISPR go from here? What's on the horizon scientifically?
The push continues on multiple fronts. Scientifically, a huge focus is still on improving precision and specificity even further.
Fewer off-target mistakes.
Exactly. And getting better at precise gene replacement, not just cutting or disabling a gene, but accurately swapping it out. Also, expanding the toolkit beyond just DNA editing.
Like what?
Well, exploring CRISPR systems that target RNA instead of DNA, which could offer more temporary, reversible kinds of edits, and continuing to refine those base editors and prime editors for ultra-precise changes.
And in terms of applications, still medicine heavy.
Therapeutic applications are still a massive driver. Advancing gene therapies for more genetic disorders, moving into precision oncology, tailoring cancer treatments based on a tumor-specific genetic makeup.
Using CRISPR to fight cancer.
Potentially, yes. And also developing the ability to do multiplex editing, changing multiple genes at the same time.
Why would you want to do that?
Well, many complex diseases, like heart disease or diabetes, aren't caused by a single gene. They involve networks of genes. Being able to modify multiple points in that network could be key to understanding and treating them.
So the science is just charging ahead.
Relentlessly.
And all this progress, I assume, just keeps feeding back into those ethical and societal debates we were just having.
Absolutely. They have to run in parallel. As the technology gets more powerful, more widespread, the discussions about responsible use, about equitable access, about managing the risks, they become even more critical. Those conversations will fundamentally shape how this technology develops and how it gets used.
This deep dive really makes it clear. CRISPR-Cas9 isn't just another lab tool. It's this incredible force that's actively reshaping our world.
No question.
I mean, from transforming farming, creating crops that can withstand harsher conditions, to offering these potentially curative treatments for devastating genetic diseases like sickle cell, like CF, the impact is just undeniable.
It really is. But as we've spent a lot of time discussing, that journey from the lab bench to the real world, it comes loaded with these really significant ethical, legal, social challenges.
Yeah, those debates around germline editing, designer babies, the potential impact on biodiversity, who gets access.
It all highlights the immense responsibility that comes with this kind of power. The power to essentially edit the blueprint of life itself.
Indeed. It feels like the core question for us as a society isn't just, can we do this with gene editing, but should we? And how?
Exactly. And that's not a question just for scientists. It needs constant informed dialogue between scientists, policymakers, ethicists, the public.
So maybe a final thought for you, our listener, as you reflect on this deep dive. Given everything we've talked about, the incredible promise and the profound risks, what ethical lines, if any, do you think should never be crossed when it comes to editing human genes or changing the environment? How do we collectively steer this incredibly powerful technology so it benefits everyone safely and fairly? Thank you so much for joining us for this deep dive into the world of CRISPR-Cas9. We really hope it's giving you a valuable shortcut to understanding this truly transformative technology.
Yeah, keep asking those critical questions, keep learning, keep exploring.